In this video, we'll look at parasite control using schistosomes as an example. So what are schistosomes? They're trematode worms, or parasitic flatworms, and they're also known as blood flukes. They look a bit like this. Unusually, for parasitic worms, there are males and females, and they have to find each other inside the human body in order to mate. When they do, the female worms produce hundreds of eggs a day. These eggs are covered in pathogen-associated molecular patterns, or PAMPs, so they stimulate a strong inflammatory response in the host. Schistosome eggs, lodged in host tissues and causing these inflammatory responses, are the main cause of disease symptoms in a schistosome infection. So how can schistosomes be controlled? Well, many parasites are very good at hiding from or subverting the host's immune responses. This means that acquired immunity may not develop, and also it's difficult to design an effective vaccine. So often, parasite control measures aim to interrupt the life cycle of the parasite. So parasite control depends on having a really good understanding of the life cycle of the parasite. Schistosomes have got a complex life cycle involving two hosts, the human and the water snail. Adult schistosomes live in the blood vessels around the bladder and/or guts, depending on the worm species. Worm eggs are then shed in urine or feces, and in the absence of sanitary waste disposal, can then contaminate fresh water. In fresh water, the eggs hatch, releasing myricidia, and these swim through the water searching for a snail host. If a snail host is found, the myricidia infect it and multiply. Then cercaria are released from the snail into the water. Cercaria can infect humans through the skin when they use the contaminated water for washing or bathing, and this completes the life cycle. Understanding the life cycle means that control measures can be implemented. For example, removal of the secondary host by the use of molluscicides on fresh waters can be effective. If there's fewer snails in the water, there will be fewer schistosomes too. To prevent cercaria from infecting humans, water should be boiled or simply left to stand for 24 hours before use. Of course, clean, safe water for everyone in the world would be better. Finally, the schistosome life cycle could be interrupted by preventing worm eggs from getting into the water. This can be accomplished by treating infected people with one of the cheap, safe anthelmintic drugs that are available to remove their infections. But due to a lack of acquired immunity, the infection is likely to return in time. Improving sanitation with the provision of toilets or latrines so that urine and feces containing eggs can't enter the water would be very effective at controlling schistosomiasis and many other diseases too. So ultimately, having a good understanding of the life cycle of a parasite can help us to understand how best to control it.